doing this presentation on the validation guide is first, why do we, so the agenda for this presentation, we will try to see why we need a validation guide for ATMPs. We'll go into the scope and the content of this guide, and then we will try to understand with few words what are ATMPs, Advanced Therapy Medicine Product. And after that, we will go into a regulatory framework for ATMPs, which is quite complicated. It's more easy across the world, but European Union and European Commission has introduced some difficulties. As everything now is based on quality risk management, we will have few slides on quality risk management. And then we will have few slides on quality by design, and we will see what are the key validation differences and challenges in ATMPs. We will go a little bit on manufacturing, supply chain, and then we will have the end of the presentation. So why do we need a validation guide dedicated to ATMPs? Validation is normally good for every product manufactured. But for ATMPs, as we have cell and gene therapy products which are issued of complex processes, the batch size is very variable. We can have one batch for one patient, we can have one batch for a few hundred, and this introduces a high complexity to the product. We have as well introduction of novel starting materials, for instance, human cells. Due to the risk of such product, it's critical to deploy a comprehensive approach of risk and identification, and we will have few words on that later. And these guys try to provide comprehensive framework for identifying, assessing, and mitigating the risk throughout the product lifecycle. At the end, we will understand that the process validation of such product, we cannot one validation for one product, but we will have to validate all the steps of the process, and this will not connect it to one product, but this will be connected to a full process. Wording clarification. We have two types of wording for cell and gene therapy. In Europe, we are using ATMPs, which stand for Advanced Therapy Medicine Product. This is used by Europe, PICS, and many other countries. It's equivalent to cell and gene therapy, which is mainly used in US. ISPE has decided to use ATMP acronym for cell and gene therapy product and advanced therapy medicine product. So in this guidance, both terms are used. Time to time we are using cell and gene, and some of the time we are using ATMPs, but this is exactly the same signification. The structure of the guide. So first we will have a section 1 which is based on principles, and we will have a general overview of what are ATMPs, and we will have a core concept and phase appropriate consideration for ATMPs. We have the regulatory framework with European Commission, EMA. We have as well FDA. We have PICS, which is an organization which make coordination in the coordination of understanding across the different inspectorate across the world. We have as well WHO. Now we have MHRA, which is UK, because we have MHRA UK has left the European Union. We have Health Canada, we have TGA, and we have all the many national competent authorities in the APAC region. Then we will have few words on quality by design, which phase the appropriate validation, and few words as well on validation, what are the key differences and challenges. And then we will have the section 2, where we'll have the phase appropriate for the validation, and we will have the chapter 6 with analytical. We will not go into all the analytical methods, but we'll make the link between analytical methods, CQAs, and validation. For the manufacturing operation, we will talk a little bit on tech transfer, but we will talk as well on CPPs, critical process parameters, and linked with CQAs, critical quality attributes. We will have some point on control contamination strategy. This will take place in the quality risk management, and we will have as well some part for raw materials and transportation and logistics. And so with that, we will cover all the content of the guide. So the scope of the document is focus on a risk-based approach. And now more and more everywhere, we are finding quality risk management based on a cell-based medicine product. We address as well tissue engineering products, which are out of the scope. For the purpose of this guide, the nomenclature of ATMP is used to refer to cell and gene therapy product, as I said earlier. We will talk about quality by design. We'll talk about quality of management. Computerized system and software are out of the scope of this guide because everything is covered by the Annex 11 in GMP. Your GMP, it's covered by the CFR Part 11 for US and some specific guidance for WHO and APAC region national competent authorities. Artificial intelligence and manufacturing innovations such as robotics are not covered neither by this guidance. These are covered by many other guidance within the ISPE's documentation. Here, I will jump over this slide. You will have here the list of where you can find other points for validation, for product validation within the ISPE documentation. Challenges and the strategies. Cell-based medicinal product present the unique challenge due to the inherent high variability of cellular starting materials. We are working with something which is more difficult, which is more with high variability, and this makes the things much more difficult to have a full validation work. And we are moving more, and we'll see that a bit later, we'll move more on continuous process verification with a high level of monitoring of the process parameters. And as well, we are using concurrent validation. We have different type of manufacturing. autologous and allogenic, which are robust control for manufacturing processes and raw material quality. This is a systematic approach that defines CQAs and their relation to therapeutic outcomes. And this is really critical. Advanced analytical tools and real-time process control enables monitoring and making adjustment during the production. And this point is really critical because we will not have, as in small molecules, the three magic batches. Once it's validated, we can run the process, and we could have always the same product. With ATMPs, it's more difficult, and we need to have more information on the product, and this will be made by monitoring. For the gene therapy products, we have viral vector, plasmids, and this face equally complex manufacturing challenges and sharing as well similarities with traditional biological medicinal products. Overview of ATMPs, cell-based advanced therapy product. We have living cells with or without ex vivo manipulation. Gene therapy we introduce into the body to treat or prevent disease. The active substance contains recombinant nucleic acid, and that will regulate, repair or replace, or delete a genetic sequence in the patient. These products are very complicated to be manufactured. They are not normally they are not always fully sterile, but the role of the manufacturing is to avoid to introduce any new contamination in the product. We can use as well tissue engineering medicines, which contains cells or tissues that have been modified so they can be used to repair, regenerate, or replace human tissues. And this is very often used now for people heavily burnt during some fires, and we can reconstitute their skin with tissue engineering product. Autologous therapies, the starting material are derived from a patient and returned to the same patient after in vitro manipulation. Allogenic therapies, the samples are derived from a healthy donor, and the final therapy can be used to treat many different patients. But when we say many different patients, we are not talking about 100,000 patients or million patients. We are talking of few thousand patients. This is a scheme who show what are the autologous therapy. So we are starting here. We are starting from the patient. We take the patient material, we manufacture, we make the dose, and then we reintroduce the dose into the patient to cure the patient. For allogenic therapies, we have the donor. We take the patient material, we manufacture, we make multiple doses, and we go to multiple patients. Regulatory framework. This is a real challenge. We do not have a full alignment across the different regions in the world. We need to have as well fast product release, and time to time, we need to inject the product to the patient before having all the results of the analysis of the product, because the analyses are too long and the shelf life of the product is too small. For instance, we will never wait 14 days for the sterility testing to be sure that the product is sterile, because 14 days is much more than the shelf life of the product. This is why we need to address during the manufacturing every information to be sure that all the process steps are under the range which has been allocated. And process validation in this context is a complex activity which needs a good regulatory framework understanding. The regulatory rules are the key point to have a validated product. For ATMPs challenges, we have small batches, time to time, one batch for one patient, and this is life-saving. We cannot fail. Short shelf life, and as this product has life-saving for the patient, we do not have the possibility to redo a failed product as the raw material is coming from the patient. Now, if we consider the organization of the framework of the regulatory, we start with what we call MRA, which is Mutual Recognition Agreement. These mutual recognition agreements are connection between two or several different regulatory agencies. For instance, in Europe, we have an MRA between FDA and Europe, and FDA has assessed each of the 28, because UK was part of European Union at that time. Each of these 28 countries has been assessed by FDA, and like that, inspection made in each country is accepted by FDA. We have many other national competent authorities. Among them, we will find ANVISA, we will find the Singapore agency, which we find China, we find many others. And if some countries find an interest of harmonization and sharing inspection results, then they can build something which is like a mutual recognition agreement. WHO do not give approval for that product, and PICS is a group of inspectors working together from now 50 plus members. China will join this group very soon, and this group of inspectors are talking and working together to understand together what are the level of understanding they need and how to apply the different regulation within this part of the work. That means as well that one point which is really on the table in the PICS at the moment is artificial intelligence, how industry can use artificial intelligence, how regulators are using artificial intelligence, and how to be sure that everything is under control. For the European regulation, we have the Annex 1, which is one of the last release document, which is really important. And then we have different parts in the GMPs. We have the part 1, which is the different chapters related to basic GMP requirement. We have the part 2, which is dedicated to active pharmaceutical ingredient. We have the part 3, where we find support document for the GMPs. We find here ICHQ9, ICHQ10, ICHQ8, and we find as well guidance related to how can we assess if a product is relevant for one facility or one dedicated facility, or it can be made in or if the facility can be address several different products. And this is a specificity for European Union and the European Commission. This is based on what we have here, the ATMPs, Advanced Therapy Medicine Products. So we have a full document. Now it is under revision, could reach around 90, 100 pages dedicated to ATMPs. So I will jump over that. The part 3, this is the document I was talking about, which is the TOX tool. Here we have all the annex, and in Europe, we can see that the Annex 2 is no longer applicable because we have only one part which is applicable for biotech product, and the other part is linked to the part 4, which is GMP for ATMPs. This is the list as well of the different documents we are using. What is interesting here is the PICS document, which is the Annex 2A, which has been developed and published in 2023, and which is a document, GMP document, on how to manufacture advanced therapy medicine product. And for the ATMPs in the PICS, in US and everywhere in the world, the Annex 1 for aseptic processes as well is connected to these advanced therapy medicine product. For Europe, the Annex 1 is not applicable to ATMPs, and in the part 4, we have a special development of some part of the Annex 1 for aseptic processes, going to this document to have sterile GMPs for ATMPs. We have as well some ISO standards for the use of general requirement for transportation of cell and cells of therapeutic use, and this is for the supply chain. And this is really critical, and we have decided to introduce some notion of validation for this part of the supply chain. QRM, the guidance has been published in May 2026. Here we have the full process for making the quality risk assessment. I invite you to go into the QRM guide. I will not go deeper in detail in this. This is what we call the full methodology to make the risk assessment, and we call that the full formality. Now, what are the points we need to consider when designing a control strategy for autologous ATMPs product? We need to take into account complexity of the starting materials. We need to take care of the process variability and flexibility, because we are monitoring, we are developing cells, and we don't know every time how the cells will grow. And we need to adapt a little bit time to time the process to have a good growth of the cells. Saying that, we need to take information from the cells grow to be sure that everything is under control. And for that, during the development of the process, we are developing what we call critical quality attributes, which give the assurance that the product is safe for the patient and will deliver the product in the right way. And connected to the CQA quality control quality attributes, we have the process monitoring and control, which are the CPPs, control process parametric. Now, one of the challenge of the QRM for ATMPs is the formality. When we run a QRM process, we can have a high level of formality and we can have a low level of formality. Low level of formality can be just application of one procedure which is well known by the company. High level of formality is to make all the process of the quality risk management for this problem. And this is based on what we know, uncertainty, the importance, and the complexity. Saying that, if we have a high level, for instance, we will need a cross-functional team. We could have with high-level SMEs, or we can have less SMEs if the level is lower. We could have the need of facilitation. That means that we could have people or SMEs who are really strong in the process of making QRM studies or QRM exercise. If the potential need for facilitation is less, we can go with only, we don't need a facilitator. And the QRM tools used as well, more the complexity is, higher is the complexity, higher is important, higher the uncertainty, more complex will be the tools used for the QRM. And here we have, we can see that the risk is decreasing as the knowledge is increasing throughout the life cycle. For instance, higher is the risk, less we will have the knowledge of the process. And if we increase our knowledge of the process, of the product, of the patient, then we will have less risk. And that means that the formality will be lower for this process with a very high knowledge. The contamination control strategy, this is something which is needed by the Annex 1, but we use it as well very, very often for the ATMPs. For instance, we are designing in a very specific way the design of the plant, the design of the processes. We try to avoid as much as possible, even if we have manual operation, as much as possible, we try to remove stuff from the contact with the cells. That's why vendors are developing more and more robotics to make this product. We are taking care as well of the materials. We are taking care of the process to be sure that the intended use of the process is to save the patient. Saying all this, we will need to go into the monitoring part. As we are capturing a lot of information, a lot of data, this will give us the opportunity to have a lot of data based on the personnel, based on the environmental monitoring, based on preventing maintenance, and this gives us a lot of information to share with the QP to be sure that we will release the product in a safe way. And all this data need to be analyzed time to time to make prevention plans, and this drive us to continuous improvement, could drive us as well to corrective action and preventive action, and this make a virtual circle for these processes. QBD, we are starting with the quality target product profile. What are the targets we want to have, and this is made during the development and the research. And then during the development, we are identifying the critical quality attributes. With the critical quality attributes, we can move into the production, and we will have for the materials what we call critical material attributes. And this is really critical because very often the material is coming from the patient. And then this will link with critical process parameters. And this will make a robust connection to be sure that the product will be released on a safe way, based on a control strategy, which is based on all the experiment which has been done during the development, and based as well on the quality risk management through the risk-based development. ICH Q9, the new revision is L1. Here we have a list of quality target product profile. I will go only in the manufacturing process, and here we start to see point we are looking now with the new processes, which are process platform. Companies are developing for some product process platform, and with this platform, they are developing new ATMPs or new cell and gene therapy product. The process platform can be validated, and respecting and using in a right way this process platform will give us a better assurance of the product. Key validation. So on the analytical part, we have the guidance from FDA, EMA. Some guidance are now developed very recently. For instance, EMA has published quality non-clinical, non-critical, and critical requirements for investigational advanced therapy medicine product. They are more focusing at the moment on products which are in clinical trials, and now this is moving as well forward with manufacturing product. And for instance, FDA has released in 2023 potential assurance to cellular and gene therapy product. This list is under development every time because ICH is developing new guidance or improving existing guidance. USP as well is developing analytical procedure during the life cycle of the product, and this is really critical. Analytical methods are connected to FDA, WHO, PICS, FDA, and EMA as well. What is critical for the cell therapies is characterization and testing, and we are testing the cell growth and how we can, if we can use them or not. Gene therapy testing as well is very specific. Potency testing for ATMPs. I will not go into the different methods, but we can find that in the different guidance. And we have as well moving into the rapid microbiome testing to be sure that we can release the product without sterility testing. For that, we need to go into the pharmacopoeias. We have accepted the rapid microbiome testing. Regulators in various countries, various areas have accepted as well the use of rapid microbial testing. Now, to be sure that everything is okay, we need to move forward with this rapid microbial testing, but the results are not exactly the same than what we have for the sterility testing. So I will jump over this one because I'm a little bit late. For the manufacturing operation, here we have simplified manufacturing process. We have autologous and cell therapy. We have the cell collection, cell isolation. We need to remove the cells we don't want to use to the cell which will be used. Then we modify the cell which has been retained, and then we expand the cells. Then we are taking the cells, harvesting them. We are doing quality assurance and testing based on all the results doing the monitoring of these operations. Then we have packaging and transportation. We need to be sure that everything is under control. And we need as well, and with that, we can go to the treatment place. And to support the process validation, we use the contamination control strategy. CCS is part of the Annex 1, could be used because it addresses sterile products, but as well non-sterile products. We can as well have a connection with aseptic process simulation, the length and the number of tests for this product, but we cannot make a lot of tests because the material is the cells of the patient. And the validation strategy has to be considered with concurrent validation and concurrent validation processes and continuous process verification. And regulators are very pushing industry going to continuous process verification because with that, they will have a better knowledge and better understanding of the processes. Cleaning is a critical point. More and more point of this of the cleaning are based on the use of single-use system. If we have equipment which are reused, we need to be sure that we have fully decontaminated this equipment before going to the autoclave and to be sure that we will not introduce some material from one patient to another one. Supply chain, the chapter is quite short. We have many interactions with patients and hospitals. The product characteristics need a strong monitoring. We have as well data sharing across the global supply chain. That means that if somebody intervenes during the supply chain, everybody in the life cycle of the product is aware of what happened in the product. When we talk as well about supply chain, we can see that with the heat of the climate, with the uncertainty in several parts in the world, this will impact the supply chain. And this could, for instance, due to the war with Ukraine, the flight between Europe and Asia has been extended from 9 hours to 14 hours. 5 hours more flight could have an impact on the product. That means we need to find other routes, other way, or to change our mind to deliver the product to the patient. Phase appropriate. We have defined here what is needed in the different phase of the development of the product. For instance, for the commercial, all analytical methods need to be validated, and we need to have continued method monitoring. And for manufacturing, we have as well for the process all these different steps. We do not have that much for commercial because many of the products are still under development, and the product with cell and gene therapy or advanced therapy medicine product are so small that they remain with the manufacturing of the late phases of manufacturing. Saying that, I'm quite to the end of this presentation. I would like to say thank you to all the contributors and the team who has been working hardly on this guidance. We have as well some individuals who have contributed in some part of this guidance. And I would like as well to give a special thank to Eric Bozenart, who was at that time the chair of the COP Advanced Cell and Gene Therapy, Advanced Therapy Medicine Product. And I would like as well to thank a special person who was the chair of this group to make this being possible. Thank you for listening, and I'm ready to answer some questions if you have any.
Thank you very much, Jean-François. So we are open to questions. So either unmute yourself to ask the question or put your question in the chat. So there is one question from David Marget. Could you explain the 3 validation batch equivalents in ATMPs and how to justify this during regulatory submission?
For my understanding, we cannot have 3 validation batches for this product because the product is related to the cells which has been delivered by the patient, and we cannot make so many batches. So we can make some theoretical validation batches, but all the product will be different depending on the material provided by the patient. This is why if we consider the Annex 15 in Europe, and this document is as well under revision, they push to continuous process verification, and they push to concurrent validation process. And with that, we avoid having the need to have 3 full validation batches, because 3 validation batches will take more cells from the patient, and generally these products are life-saving, and we cannot take that much cells from the patient. This is why this we need to think in a different way. We need to think out of the box, and we need to go into the quality risk management process and see what is the level of knowledge we have, and how and where we need and where we can make the best improvement of that.
Thanks,
But
John.
for sure, regulators want to push us, industry, to have more monitoring in our processes, which are complex, and to give more information and release with more data the product.
Yeah, thanks, Jean-François. There's another question from Renee. What's the difference between CQA and CPP? Does CQA relate to quality testing? Does CPP relate to conditions such as temperature, CO2, etc.?
Yeah, so this has to be made during the development of your product. during the development of your product, you are in the research part, you are trying to have the CQA, which are the parameters, the analytical parameters, which will give you the best way to release a safe product for the patient. Now we are monitoring, we are making manufacturing. And for the manufacturing, we cannot analyze the cells, but we have parameters during the manufacturing which will give us the information that at the end of the process, if this parameter is between the range, we meet the CQA, that means the critical quality attributes. This is why we need to make a link between the CPPs, critical process parameters, and the CQAs, which are the critical quality attributes. The critical attributes will give us the way to release the product. The CPPs will give us the information during the manufacturing process that the process and the product is good and will meet the CQAs.
Yeah.
And this is something which is really critical because few months ago, EMA has released a Q&A document which is based, and this document was to try to answer a question: can we, and what happens if we release a product with an out of specification during the manufacturing? And this is critical because very often when we understand that we had somewhere an out of specification, the product has already been injected to the patient. And this is why we need to have a good process monitoring, and maybe artificial intelligence in the near future will give us the way to gather all and to analyze all the information coming in our hand to give us a way to give the right decision at the right time.
Yeah. Yeah, I think that future sounds exciting, right? On the other hand, AI for regulatory decision making or regulatory relevant decision making is going to be a huge topic that the industry needs to address in the near future collectively. So I wonder if there's any question among the other audience. Do feel free to unmute yourself and ask the question. We do have a couple of minutes left. So perhaps I can ask a follow-up question, right, to the magic three, the three validation batches. So for autologous CAR T, cell therapy manufacturing, I think it's really common to use healthy donor starting materials to qualify your process to make sure, the parameters are set in a scientifically sound way, resulting in the process to perform as expected. So I just wanted to ask your real-world experience, Jean-François. So healthy donor batches as part of process qualification/validation, how meaningful are they?
So I think for autologous product where one patient gave healthy cells, and we need to go to the patient. This could be made with, I will say, in a theory, could go into the three validation batches. Now we are in a part where we are in R&D. We have, even if we have a healthy patient, at the end we have patient waiting to receive this product. And if we lose time to do a real 3 validation batches, maybe some patient will lose some
Mhm.
chance
Yeah.
for life. And this is based on quality of risk management. Do we have enough product? How can we make it? And maybe the way to make it is, can we reduce the number of patients at the end, um, to be sure that all will receive the product in time? Because we are in a part where for the patient, our products are really life-saving.
Hmm.
And this
Yeah.
makes the things really, really, really complicated.
Yeah. Absolutely. Yeah. So perhaps one final question. How do we select what type of process validation that is suitable for allogeneic CAR T manufacturing?
I do not have a ready-to-use answer. For me, the best way is to go into the quality risk management and to go into the Annex 15.
Hmm.
I think that several so in the ICH Q9 documentation within the ICH website, they have produced a lot of training materials, and maybe we can find some answers in this way.
Yeah.
We can find as well answers in some other ISP guidance, which are, I think a specific guidance on, we have a specific guidance on allogenic therapies. And with that, we can find some other answers as well.
Yeah.
And maybe in the near future, we could use, uh, if it will exist, an AI tool going into all the ISP documentation to find some solution or some answers for these questions.
Yeah.
But for sure,
Indeed.
this
Yeah.
is really critical.
Yeah, we have many hidden gems, right, among ISPE body of knowledge, guidance documents, and various literature, including a pharmaceutical engineering magazine. So time is limited. I think that's it for today. Before we close, I would like to thank Jean-François Duliere again for your wonderful presentation. Thank the audience for your active participation. Just a reminder, this presentation is recorded and will be put at ISPE Singapore affiliate YouTube channel. So do check out our YouTube channel to replay if this is very useful for you, and check out previous webinar as well. And let's keep in touch. We definitely look forward to your participation again in our future webinars, perhaps allogeneic cell manufacturing as one of the future topics. So with that,
Okay.
thank you, everyone. I wish you a good day. Thank you very much, Jean-François.